Hello, good morning to dear students and those are interested in biological system. It is my pleasure to once again to discuss and illustrate a biological cell in the context of by taking a very simple illustration and the example of writing pens that will represent proteins in a biological cell. So in a very simple way, I will try to make a comparison and also the similarities what exist between the the routinely used in our life, that is writing pens, and also on the other side the proteins, that is the key aspect of any biological system, whether the cell as prokaryotes or whether the cell as eukaryotes. All right, my dear friend, if you look at interestingly, P alphabet, that is for pen, and P alphabet, that is for protein. So since a long time I had certain thought process so that we can make a illustration between these two word, that is pen, that is used as writing pens, and that is P for protein in a biological cell system. All right. So always a question comes to my mind. Okay, and all of us know, as a student, even those are in the school, high school, definitely writing pens can be of different sizes, composition. It can exist across the globe in different number. If you look at one single cell here, let me to use also the annotation. Okay, so if you look at the biological cell here, and if you start from the ECM, that is extracellular matrix, then plasma membrane, then intracellular fluid system, and then the nucleus. P, that is for protein, exists in a various composition, definitely dictated by the amino acid sequences, but they are present across all the compartment of a biological cell system, inside nucleus, inside mitochondria, outside the membrane, etc. So it looks very clear similarities between those writing pens. If you look at here, it can be of different sizes, different color, different texture, composition, blah blah, etc. The another very important what I noticed, the writing pens can write in different colors with different efficiency or the speed. And if you look at, my dear friend, that is protein across whether in the ECM or membrane or ICF or the nucleus, they can also perform various functions, whether in the form of replication, transcription, metabolic function, whether inside the or on the plasma membrane to work as a receptors. So the varied functions are attributed by the nature, and that is for even one biological cell we can conclude. And also the these proteins, whether in the different compartment of the cell, they have the various different efficiency, the way it works, and that is why I thought it's very interesting. You can say illustration between one, that is writing pens, and the proteins in terms of difference in their ability to perform functions, whether in the form of different color in the cell, you can say different metabolic activities, etc. All right. Another very interesting point is uh, it's I would like to show and display here. If you look at these two, one pen is here and other pen is here. So if you look at This pen is going to have the cap, and this is the pen where we are going to write here. So if the pen is going to be with the cap here, the pen cannot write. Yes or no? And to write by the help of this particular pen, we have to go for removing the cap. So my dear friend, that is what I understood. In a biological cell system, wherever proteins are there, some proteins, they have the almost same identical mechanism where what I propose, the chemical caps are there. So most of the protein, they are not working always. They are not doing the job. So most of the protein, they are capped structure by the help of chemical caps. And to allow the function of those protein, these chemical caps has to be either removed or chemically changed. That is what I understand. Interestingly, in comparison to this cap where the pen is going to work with the help of cap, other pen, if you look at here this way, there is no cap. Okay. So this protein, see this writing pens is going to have another mechanism. By pressing, we can allow so that pen can write. So similarly, our proteins in biological system, they have almost a similar kind of mechanism where inherent or internal structures are there in the protein that facilitate for inhibition or activation of the protein. That's what I thought I should share here. These are very interesting similarities between these writing pens and the proteins in a biological cell. All right. Another very interesting I would like to share here. Look at. So these are the two different structure of the pen design. So this is the cap of one pen. This is the cap of other pen. So this cap cannot fit to this particular pen system, and vice versa. You can see it cannot. So likewise, our biological cell, and where the thousands of proteins are there, so they are created in such a unique design. Technically or scientifically, we say primary, secondary, tertiary, and quaternary structure in a very simplified manner if you look at the student understanding. So these are like different cap-like structures are given for each proteins. So it cannot fit to the other. So in other way, you can also we know that the catalytic mechanism of the enzymes. So almost it is like the same concept. So one cap of the one pen cannot fit to the uh, other pen structure. That is the whole mechanism. Another very interesting point is, and it is my responsibility to explain here, in a biological cell, in one single cell, or even in the nature, if you look at the writing pens, one writing pen of the same design can exist in multiple copies. Look at here, two copies, or it can be billions. Likewise, in our biological cell system, one protein of unique design or the chemistry, they can exist in multiple copies, and that depends upon the type of the protein. All right. Another very interesting that is my interest is here. Either writing pens or proteins in a biological cell, they are not immortal. They are not going to exist forever once it is created. That means that can be degraded, that can be broken, or that can be recycled. So likewise, if you look at the writing pens, so these are not going to be stay forever. So it has to be degraded, it has to be recycled, it has to be broken. So similarly, the proteins in our biological system, they are degraded, recycled, and that is how the nature is going to work, and that is how our cell system is going to work. Another very important point is, if this pen is going to write something, the written content is also not permanent or forever. The written content is also going to be erased. So likewise, if you look at the writing pens, yes, erasers are there. So in our biological cell system, similarly, these chemical languages, those are going to be written by these proteins that are going to be erased from time to time, and depending upon the different mechanism within the cell system. That means erase or to erase that particular chemical language in a biological cell is very, very thoughtful process. And that is what also what I think similarity between these writing pens and uh, proteins in a biological cell. All right. One very important thing we have to consider here, if you look at all the, this is the one type of the pen, this is what I say, uh, another type. Okay. Irrespective of the different color, size, composition, etc., there the majority of the function is to write. Yes, sir. So similarly, in our biological cell system, proteins may be of different chemistry, different composition of amino acids, but the basic functions are the same. Or one more important, whether the writing pens are used one part of the globe or other part, functions are the same. Similarly, these proteins, whether it is for prokaryote or eukaryotic cell, the functions are almost, basic functions are almost identical. Another very important, it's very interesting that these writing pens, once it is created, they cannot create its own copy. So this writing pen cannot create its own copy by itself. So likewise, the proteins, once it is created in a biological cell, protein cannot create its own copy. Rather than protein has to be manufactured or translated from, like in biological cell we know that is DNA, then RNA, and then to the protein system. Means the self copy cannot be created by protein itself. That is what one of the you can say uh, hallmark of this particular system, or the protein as a system. Another very important point is, if you look at this writing pens, this one, this has the capability of writing itself, but some of the writing pens can have the capability of both writers and as erasers. So likewise, in our biological cell system, naturally some of the proteins are present, those have the responsibility of writing the chemical languages, and also the responsibility of erasing those chemical languages. And if you know the uh, epigenetic phenomenon, that is one of the landmark example where the chemical languages on the DNA sequences, that is chromatin, or also to the chromatin proteins, are going to be written and then erased. So the proteins are already in existence. So that's why also it is very interesting comparison between the writing pens and some of the proteins, those are working inside the biological cell system. Okay. Interesting, I also noticed. So nowadays, some of the writing pens, they are also equipped with recorder facilities. If you look at here, some of the pens they have. So these recorder facilities, surprisingly, if I look at, and even we proposed way back, that some of the protein in a biological cell system, they have the capability of as to serve as a molecular recorders. They can record various events within the cell, like uh, transcriptional events or chromatin remodeling events. That is very also interesting, and that it connects the dot between the similarities of writing pens and proteins in a biological cell system. Another I also noticed, so some of the writing pens, they can also attain the properties of as a highlighters. It can highlight certain languages or the written content. So I looked into the literature and it looks very true. So some of the protein in a biological system, they have the properties of fluorescent proteins naturally. And these fluorescent protein, what I think, they are almost close to the same function of like writing pen system where the fluorescent, I mean the highlighting properties are there. If you look at here. And last point is, some of the writing pens, new technology pen, they can also read, record, etc. So likewise in our biological cell system, already it is discovered. So some of the proteins, they can serve as molecular readers as a part of epigenetic phenomena. That's great. So altogether, if you look at, the comparison between the writing pens and proteins in a biological cell system, almost it is close. And I do not, and also the alphabet P for pen and P for protein. It looks very interesting to me, and that is what I made efforts to summarize. And also, it will give you better understanding about the protein, about the biological cell, and how the different types of protein in a biological cell, they are doing wonder jobs. Thank you very much for listening. Drop by drop, please keep learning and keep your imagination in a very vibrant mode. Thank you very much.